Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my lecture. So, in this lecture today, we will discuss the different types of RNAs. As you know, there are three major types of RNAs, as these are the ribosomal RNA, transfer RNA, and messenger RNA. So, basically, what are these RNAs? These RNA is basically are the unbranched polymeric molecules that are composed of nucleoside monophosphate, and all of you know that these monomers are joined together by 3' to 5' phosphodiester linkages or phosphodiester bonds. So they are differ from the DNA in several ways, as these are the unbranched molecules and they contain uracil instead of thymine, and these are normally smaller than DNAs. Today we are going to discuss the RNA structure and the various types of the RNAs. So there are basically many types of the RNAs, including transfer RNAs, ribosomal RNAs, heteronuclear RNAs, small RNAs. But the major types include are the transfer RNA, ribosomal RNA, and the messenger RNA. So today specifically we will discuss the messenger RNA. So basically, messenger RNA is the connecting link between the DNA and the protein synthesis. As you know that the RNA are basically the non-genetic molecules. These are involved in the protein synthesis. However, there are these can be act as a genetic material for these certain viruses. ठीक है. So if this is the central dogma of the life where the DNA is converted into the RNA, and then this RNA is translated into the protein. So this is how the information is going to get transferred from the DNA to RNA. So this RNA will read the specific sequence in the DNA. You know that DNA stores all sort of genetic information that is present in the genes, in the GNA, in DNA, in the form of the coding sequences. So this messenger RNA is going to copy that particular sequence and again translate this particular sequence into the proteins. So these are the transfer RNAs. They are basically involved in the transfer of the amino acids from the cytoplasm to the site where the proteins are actually being synthesized. And this ribosomal RNA molecules, they basically assist in the synthesis of the proteins. So let's talk specifically about the messenger RNAs. So what are these messenger RNAs? As I have already told you that this messenger RNA is going to be the connecting link. Basically, it is going to link the particular DNA, that is the coding sequence, and its expression product, that is the protein. Normally, the expression product can be RNA as well as protein, but this is only the messenger RNA that is going to be translated into the protein. So this particular process, basically the copying process, during which this DNA strand is basically serve as a template for this synthesis of the RNA. This particular process is the transcription, and then again when this messenger RNA is going to translate into its product, its functional form, that is the protein, and this process is the translation. So what we can say that this molecule, that is this messenger RNA molecule, is helping in the transfer of the information from this DNA to the ribosomes for the synthesis of the proteins. As you know, the protein synthesis is basically takes place in the ribosomes, in the cytoplasm with the help of the ribosomes. So these messenger RNA molecules, basically these are first synthesized in the nucleus. So very first thing that is synthesized in the nucleus from the DNA is not the mature messenger RNA. So the very first thing is the heteronucleus. You can say heterogeneous nuclear RNA. So this is the you can say the primary transcript that is going to be synthesized from the DNA. So it is having everything that is present on the DNA, having all those introns and exons, everything that is present in the DNA is present in this particular type of the RNA. And then this heteronuclear RNA is going to be the precursor for the synthesis of the mature messenger RNA. Now, this is the outline for the process that is going to be happen in the nucleus. So, what are these E1? So, here you can see this is the particular DNA or particular gene that is comprised containing that specific sequence, specific codes, coding sequence that is storing the basically genetic information. And this is the primary transcript that is synthesized from the DNA in the inside of the nucleus. So it contains the coding region as well as the non-coding regions. So these regions in the blue, these are basically representing the exons. Exons are known as the coding portion of the coding region that is actually going to translate into the proteins. So these regions in the black, these are like I1 and I2. These are the non-coding regions. Non-coding regions जो होती हैं that basically do not code for anything. The non-coding regions will not translate into the proteins. जो प्रोटीन बननी होती है वो सिर्फ हमारी जो कोडिंग रीजन्स है मैसेंजर आरएनए में वो वहां से प्रोटीन्स बनती हैं. ओके सो basically this heteronuclear RNA that is containing the introns as well as exons. So here the special players will come. These are the endonucleases that basically that cut the nucleotides or break the bonds between the nucleotides. They will cut at their boundaries. Like here, these are the exon 1, intron 1, then exon 2, intron 2. यहाँ पर आपने देखा है कि इसकी बाउंड्रीज पर इसने कट लगा दिए हैं. Now there will be a special process which basically involves the removal of all the introns. So at this step all the introns, that means all the non-coding regions, जितने भी non-coding regions है ये सब यहाँ से remove हो जाएंगे. और इस process को हम क्या कहते हैं? This particular process is the splicing. जब ये नॉन कोडिंग रीजन्स रिमूव हो जाएंगे और जितने भी कोडिंग रीजन्स हैं जिनको हम एक्सॉन्स कह रहे हैं ये सब हम ज्वाइन हो जाएंगे विद द हेल्प ऑफ द स्पेशल एंजाइम्स लाइगेजेस. तो इस पूरे प्रोसेस को हम स्प्लाइसिंग कहते हैं और ये प्रोसेस इतना सिंपल नहीं है जैसा कि इस पर्टिकुलर स्टेप में बताया गया है. दिस इज जस्ट an outline for this particular process. So what is going to be happen that first of all we have the sequence, we have the information coding sequence in the DNA जिस और इसके बाद जो प्राइमरी ट्रांसक्रिप्ट बनेगा उसको हम कहेंगे HNRNA. और दिस HNRNA इस बेसिकली क्या है? ये सर्व कर रहा है, इज सर्विंग एज अ precursor for the synthesis of the mature messenger RNA. So in the process of the splicing, जितने भी introns हैं, they will remove और जितने भी all the exons, they will join with the help of another players that are the ligases. Now, what happened? This particular molecule, जिसको हम कह रहे हैं, this is the messenger RNA molecule. It's you can say it's a highly unstable molecule. So what we need to do, this molecule is need to get stabilized. So how it is going to get stabilized? It will stabilize by the addition of a cap and a tail. ठीक है. Now, what is the capping and the tailing? So, in case of eukaryotic messenger RNA, this particular messenger RNA is basically capped at this terminal. इसके 5 prime terminal पे there will be addition of a 7-methyl guanosine triphosphate. ठीक है. This 7-methyl guanosine triphosphate. So let me write here that This particular molecule that is used for capping is the this is the 7 methyl guanosine ट्राइफॉस्फेट। ठीक है, ये मॉलिक्यूल जो है दैट इज यूज्ड फॉर द कैपिंग। और ये किस टर्मिनल पर आके ज्वाइन करेगा, यहाँ पर इसके 5 प्राइम एंड पर। ठीक है, और इसके 3 प्राइम एंड पर देयर विल बी एडिशन ऑफ द पोली ए टेल। व्हाट इज दिस पोली ए टेल? ये जो पोली ए टेल है, दिस इज बेसिकली द, ओके लेट मी राइट हियर। सो ये पोली ए टेल जो है दैट इज बेसिकली दी polymer polymer of what it is the polymer of the adenylate residues। ठीक है, polymer of the adenylate residues। That is these are the adenosine mono phosphates। so यहाँ पर जो एडिशन होगी एडिनिलेट रेसिड्यूज की, और इसीलिए इसको हम कह रहे हैं दिस इज द पोली ए टेल। नॉर्मली दीज आर यू कैन से देयर नंबर बेसिकली from 20 to almost 250 residues will add at its 3 prime terminal of the messenger RNA। And what is the purpose of this capping and the tailing? एक तो ये कि कैपिंग और टेलिंग ने इस मैसेंजर आरएनए को एक स्टेबिलिटी प्रोवाइड करनी है। इसके अलावा जो सेल में नंबर ऑफ यू कैन से जो एक्सोन्यूक्लियसेस होती हैं, ठीक है, there are exonucleases at the 5 prime end and at its three and for the 3 prime end। तो ये एक्सोन्यूक्लियसेस का क्या पर्पस होता है? जो 5 प्राइम एक्सोन्यूक्लियसेस होती हैं वो यहाँ पर कट लगाती हैं। ठीक है, इसी तरह जो एक्सोन्यूक्लियसेस के अटैक से सिक्योर करने के लिए भी दीज आर दे हैव रोल फॉर बेसिकली इसका रोल किस में होगा कि ये क्या करेंगे, जो अटैक होगा फ्रॉम दिस एक्सोन्यूक्लियसिस उससे इसको प्रिवेंट करेंगी। ठीक है, नाउ देयर आर अगेन we take the spotlight। ठीक है, so here finally you can see that this is the mature messenger RNA। ठीक है, so basically this messenger RNA is carrying the information from particular gene। ठीक है, now every this messenger RNA that is the I'm talking about the mature messenger RNA, उसके start में there will be the UTR, that is that are the untranslated region, जो कि 5 प्राइम टर्मिनल की तरफ होंगी तो these are known as the 5 प्राइम UTRs। These are the 5 प्राइम untranslated regions। So these are the 5 prime untranslated regions। and again there are 3 prime UTRs। मतलब untranslated regions का क्या पर्पस है? मतलब ये कि जो ये पर्टिकुलर regions हैं इन मैसेंजर आरएनए दे आर नॉट गोइंग टू ट्रांसलेट इनटू द प्रोटीन्स। जितनी सीक्वेंसेस इस रीजन में आएंगी ये आगे कोई भी फंक्शनल प्रोटीन्स नहीं बनाएंगी। तो इसको हम इसीलिए कह रहे हैं दीज आर दी UTRs, दीज आर दी अनट्रांसलेटेड रीजन्स। देन एवरी मैसेंजर आरएनए के स्टार्ट में हमेशा एक स्टार्ट कोडॉन होगा। ठीक है, जो कि हमेशा होता है AUG। देन एवरी मैसेंजर आरएनए विल हैव अ स्टॉप कोडॉन, कि ये पर्टिकुलर वो रीजन है जहां से प्रोटीन सिंथेसिस के लिए जो कोडिंग सीक्वेंस है उसको हम कंसीडर करते हैं इन द पर्टिकुलर प्रोसेस ऑफ द स्पेसिफिक प्रोसेस ऑफ द ट्रांसलेशन। सो स्टॉप कोडॉन्स आर ऑफ 3 टाइप्स। दीज आर द UAA, UAG and UGA। so this central portion that is the in blue, this is the structural gene area। this particular area is going to translate into the specific proteins। ठीक है, now if we look at the typical structure of the messenger RNA having this 5 prime cap, having UTRs, start codons and stop codons, this particular RNA now it will move out of the nucleus and into the cytoplasm, and this will help in the process of the translation for the synthesis of the proteins. So this is the characteristic features of for the mature messenger RNA. Now, if we look at the structure of the typical mature messenger RNA, then these include its the 5 prime cap, that is the 7-methyl guanosine triphosphate at its 5 prime terminal. Then we have 5 prime UTRs, as these are the untranslated regions at its 5 prime end. Then we have coding region, जिसने ultimately आके translate होना है into the proteins. Then we have UTRs and then this poly A tail. Now basically if the messenger RNA, basically जो gene है उसको हम क्या कहते हैं, this is the you can say is the is equal to the gene. So if we write that one cistron, see one cistron basically is equals to the one gene. So if this messenger RNA that is basically carrying the information from one particular cistron, one gene, तो उसको हम कहेंगे this is the monocistronic. And if this messenger RNA carrying the information from more than one gene, तो उसको हम कहेंगे polycistronic. पोलीसिस्ट्रॉनिक, ठीक है। तो बेसिकली सिस्ट्रॉन हम किसको कह रहे हैं, सिस्ट्रॉन बेसिकली हम कह रहे हैं कि वन सिस्ट्रॉन इज इक्वल्स टू दी वन जीन. So this is the whole process. So if we look at the major types of the RNAs and their composition within the cell, then there are basically several different types of RNAs that are present inside of the cell. They include the nucleolar snRNAs, microRNAs, ठीक है, that we will discuss in our future lectures. Now, the major types of the RNAs that are involved in the synthesis of the proteins, these include the ribosomal RNAs, messenger RNAs, and transfer RNAs. So this messenger RNA is basically make up the 5% of the total RNA in the cell, because this actual messenger RNA जो है, this is going to translate into the proteins, जिससे हमारे जो एक्चुअल जो प्रोडक्ट्स हैं वो बनेंगे. एंड दिस राइबोसोमल आरएनए इज ऑलमोस्ट 80% present in the cell and the transfer RNA is the 15% of the total RNA present in the cell. So these three types of the RNAs, these are required for the synthesis of the proteins.